Welcome to Cellulin Science for Everyone. My name is Roger. I'm a scientist living in Montreal, and this is my channel where we talk every day about science that may interest you. CRISPR. When we hear the word CRISPR, most of us think about genetic editing. In other words, modifying the genetic information in organisms. In the news, we can hear how CRISPR could be the solution in the future to genetic diseases. However, CRISPR can also be used to answer questions regarding how space exploration will affect our bodies. As we travel beyond Earth, our DNA can be exposed to damage caused by ionizing radiation. On Earth, some workers are exposed to ionizing radiation sources. Workers like radiologists, nuclear power plant workers, uranium miners. But in space, ionizing radiation will be much stronger than the sources that we are currently being exposed to here on Earth. But are we not exposed to cosmic radiation on Earth? Well, the Earth's magnetic field protects us from the powerful natural sources of ionizing radiation in the universe. Sources like solar proton events and galactic cosmic rays from cosmic sources. When humanity will travel through the universe, we will not have the protection of the Earth's magnetospheres. And among other problems, ionizing radiation can damage the DNA, producing what is called double-strand breaks. If you imagine the DNA as two parallel wires, double-stranded breaks will cut both wires. And these can lead to mutation, genetic instability, and problems that we want to study before we explore the universe. A way to reproduce the double-stranded break in a controlled fashion is using CRISPR. This genetic tool that many compare to scissors cut the DNA at a really specific region of our choice. This cut can be repaired in two ways. The first, let's call it a crappy mechanism called non-homologous end joining, during which insertions or deletions may be added at the DNA breaking site. So we can introduce errors, and we know errors in the DNA can be tricky. The second mechanism is a fancy and precise mechanism called homologous recombination, in which the DNA sequence often remains unchanged. During homologous recombination, a homologous DNA sequence, so a DNA sequence that is the same, is used as a template for repairing the DNA. Therefore, the DNA that has been cut remains unchanged. Previous works suggest that the space condition may impact the choice of DNA repair pathways. Well, we know which is the ratio between the two mechanisms, between homologous recombination and non-homologous end joining on Earth. Now, we just need to calculate if this ratio has changed on space. And how are we going to do it? Using CRISPR on the International Space Station. So far, thanks to a paper published recently, we know that scientists have identified the conditions to perform the CRISPR experiments on the International Space Station. Now we just need to see which are the results. Thanks for watching this video. Please like and subscribe if you think that we deserve it, and see you next time.